It is my great pleasure to introduce Professor Joanne Lemieux, who is here at the University of Alberta as a professor. She did her bachelor's degree in science at Dalhousie University, where she also did a master's, and then she got her PhD at New York University, my old stomping grounds. She did a postdoctoral fellowship in biochemistry and structural biology here at the University of Alberta before joining as a faculty member. And she's currently a professor at the University of Alberta. She is the CRC Emeritus in Protein Structure and Function. She got a CIHR new investigator as a new investigator. She's the current director of the Membrane Protein Disease Research Group and the scientific director of the new Prairie Home Pandemic Preparedness Center. So she's going to tell us about her work with antiviral drug development, targeting the SARS-CoV-2 main protease and viral proteins' influence on galectins. So with that, take it away, Joanne. You need a clicker. There's a clicker.
It is on. I'll put it a little closer to my lapel a little bit more. Okay, great. So yes, I'm Joanne Lemieux. I will have to make a disclaimer. I'm not related to Ray Lemieux, so it's a separate family. But I'm really happy to be talking here today. Again, maybe $1 million baby story, because we're going to do a combination of glycobiology and kind of chemistry and antiviral drug design. And as Lara mentioned too, I also help the scientific direction for the Prairie Hub for Pandemic Preparedness, which is led out of the University of Alberta here. So here's just to start off a picture of my lab, and some of the work that I'm going to talk about today was really spearheaded by Amelia and Jimmy in the lab, as well as our research associate Elena. And our lab really focuses on proteases. So we have a membrane protease family that we work on related to pathogenic bacteria. And then we also work on viral proteases. So we'll talk about that, the viral protease story today. Let's see. Okay, so many of us are aware of this, the coronavirus life cycle. And one of the reasons I like this slide is, oops, sorry, I'm gonna get used to this clicker. Yep. Is that it really talks about, the slide really tells us about the different mechanisms of entry, so either by receptor binding or endocytosis, and then we have fusion and then primary translation of the positive strand of the RNA for the virus, which is then cleaved by proteases. And in red, we have all of these different mechanisms for inhibition. So once the viral protease is, once the viral polyprotein is translated, it's then cleaved by the viral protease to form the little pieces that will come together to form the replication machinery to make new virus particles. Then during viral RNA synthesis and assembly, you then get viral maturation and virus release. So this is a well-known pathway with all of the steps here. And in red are different possibilities for inhibition. And what I'm going to talk about today is our lab's effort to look at the viral protease for SARS-CoV-2, how we've developed inhibitors, and how they also influence galectin inflammation response proteins in the host and how the host responds to infection. So as I mentioned, the SARS-CoV-2 genome is translated into a long single polypeptide. And within this peptide, we have structural proteins. So many of us are aware of the spike protein, but then there's also these non-structural proteins, and they play primary roles in the maturation and assembly of a new virus particle. And within this polyprotein, there's actually two proteases that are encoded. One is the papain-like protease, found here, and papain protease will cut 3 sections within this long polyprotein, but then the C3L protease, also called the main protease, will cut 11 sites along this polyprotein. And so this is one of the reasons that this protease is such a highly regarded target, because it is one of the main proteases that is responsible for releasing the factors that will make new virus particles. So traditionally, over time, we know that many proteases have been targeted and used clinically for both cardiovascular and diabetes, as well as antiviral countermeasures. So traditionally, HIV protease was really integral in preventing the infection and spread of HIV, as well as hepatitis C protease. I will mention that Pfizer last year released Paxlovid. So Paxlovid contains nirmatrelvir, which is an inhibitor for the main protease, but it also contains another additive, ritonavir. So ritonavir is a pharmacokinetic booster to make this antiviral drug a bit more effective. However, for some people who are on other medications, this would affect the metabolism of the medications they're on. So this Paxlovid is really formulated not for those immunocompromised or those people who tend to be the aged population where they will need potentially to have the other medications still be taken. So our lab is really working towards developing the next generation inhibitors. Now, besides the viral protease cleaving the viral polyprotein, it's also been recognized over the last few years how the proteases found in the SARS-CoV-2 affect the cleavage of host targets and how this affects the host response to infection. So many people have worked on this. Here I'm highlighting one from Chris Overall's group at University of British Columbia. So Isabel, his lead postdoc who worked on this project, they identified 101 human proteins that were cleaved by SARS-CoV-2 main protease. And some were presented as they demonstrated that they were bona fide substrates. Now, what was important is that they found that SARS-CoV-2 played a role in xenography pathway. And so this is the host response to respond to infection. And so this includes galectin-8. So galectin-8 binds glycans. And importantly, they showed in this paper that the galectin-8 was binding to the glycans found on the spike protein. And then lastly, they showed that the main protease found in SARS-CoV-2 also cleaved galectin, again, altering the host's response. Getting some feedback, so I'm just gonna... So a bit more on this. So what is galectin-8? So galectin-8 is a carbohydrate recognition protein. It's actually a heterodimer. So there's two carbohydrate recognition domains with a linker in between. And the overall group showed that the linker was cleaved in between. And here they show a model of the GAL8 bound to the main protease. Now, GAL8 is not the only GAL, as I'm sure this audience knows. So there's different flavors of galectin. So some are homodimers containing the same carbohydrate recognition domain. Some are heterodimers, so they have different carbohydrate recognition domains. And then we have this interesting galectin-3, which has a single carbohydrate recognition domain. But what we do know is that all of these tend to bind disaccharides, such as glycnac and lacnac. And we're really starting to scratch the surface and understand how galectins play a role in infection and immunity. So they're known to play roles in various aspects in the cell, including mRNA splicing, apoptosis, cell cycle, as well as cancer. So they're upregulated in lots of cancers, and then also in the immune response. So in the immune response, here's a figure from a paper that was recently published just last year about the role of galectins in immunity and infection. And the model is thought that upon the exposure of a cell to a microbe or virus, that there would be some disruption in the lysosomal compartment. And this disruption would expose glycans. And these would be recognized either by the carbohydrate recognition domains or singles. But we do know that galectin-8 plays a role in this recognition. And this will then target that lysosomal vesicle for degradation, so autophagy, also known as xenography. And so this is a way for the host to help defend itself against an attack against some virus. Now, as I mentioned, the galectins can be found in the nucleus, cytoplasm, extracellular space. They play lots of diverse roles in the cell. But today, I'm just going to talk about galectin-8 and 9. And so we do know some structural work. So my lab is an X-ray crystallography lab. And here's the crystal structure of galectin-8. with the carbohydrate recognition domains and a very flexible linker in between. So we know that that flexible linker is cleaved by the main protease. And for the main protease, and all proteases for that matter, they typically have a three-dimensional shape. And this shape specifically will recognize a substrate. And we have a nomenclature that's here. So typically in the protease, we have these S sites called subsites. And so in between where it recognizes the substrate with the respective P sites, the P sites will fit into the S sites. And P stands for product here, and we have cleavage that occurs at this cis-al bond. And that's also kind of shown here. So if you recall, the peptide bond has this chemistry, and there's the cleavage that occurs here at the cis-al bond. And so while we're developing inhibitors, we can use this specificity to inform us on how to precisely have a strong affinity for this recognition site. Now, fortunately for us, SARS-CoV-2 has a strict requirement for glutamine in the P1 position, and this is one of the things that we've been able to take advantage of. If we go just a little upstream to the P2 position, we see leucine is also highly preferred. So this would be in these two positions here. And so if we go to the catalytic cycle of proteases and how they cleave peptide bonds, this is something that can be taken advantage of. Now, where my talk is going to go today is over the past few years, we've obviously recognized that we've had various waves of COVID infection from the first wave that occurred late December to April. We have our first wave, the wild type to first wave. Then we had the beta, alpha, delta, as well as gamma and Omicron. And so with every wave, you've heard a lot in the community about changes in spike protein and about how the vaccination will be, recognizing the new strains. So it's my advertisement to get vaccinated, because I did attend recently a talk about T-cell memory, and the evidence is pretty clear that you need a new vaccine in order to be protected. But so this is where we're going to talk about. We're going to talk about how Paxlovid was introduced around here. And then one of the main things that we want to ask is whether or not, as the virus evolves, whether or not antivirals remain potent, and whether or not the change in the host remains the same. So often you can have resistance that occurs because of a treatment. So this occurred, of course, with HIV. So there was resistance to the protease inhibitors, and combination therapy was needed. But sometimes you can also acquire resistance just by a variant of concern. And so this is something we wanted to monitor, whether or not these changes in SARS-CoV-2 over the last couple of years, whether or not they altered our ability to generate potent inhibitors, and if the host response changed. So I will introduce the team that I work with. So I'm really just a representative in this really amazing group here at the University of Alberta. We work with two chemistry groups, John Vetters in the Department of Chemistry and Jim Nyman at the Applied Virology Institute. Then we have the biochemistry arm, where we do lots of enzymology and crystallography. And we have our virology arm led by Lauren Terrell and Michael Joyce. And over the past couple of years, our team has published quite a few papers on different inhibitors. And I'm not going to show you all that inhibitor study, but I'll give you a little taste of it, and just then how that affects the galectin host response. So here, I'd like to have this slide in because these are some inhibitors developed in the 90s, 1999. So it really tells you, again, a lot of the Paxlovid inhibitors are developed against our knowledge in the past. So many of them have different warheads. So these are the electrophilic warheads that will be attacked by the nucleophile. And often there's certain groups that recognize. But you can see here I have that P1, P2, P3 nomenclature that we discussed before. So we have different warheads like esters, we have aldehydes, and we have acyl oxymethylketone type of warheads. Interestingly, this is an inhibitor developed against SARS-CoV-1 outbreak that occurred in 2003. So how our pipeline works is we have different inhibitors that are developed over the past couple of years. We've worked with over 150 inhibitors. Our lab will do enzymatic assays to measure the inhibitory concentration, followed by plaque reduction assay in the Terrell lab. And then we do X-ray crystallography to inform on the inhibitor development, and we'll feed back to generating new inhibitors. One of the first things we did, and I always like to highlight this, is the change between SARS-CoV-1 and SARS-CoV-2. So we purified both the protease from SARS-CoV-1, that was the outbreak in 2003, to SARS-CoV-2, and this is the wild type version. And when we did the enzymatic analysis looking at the catalytic parameters, so this would be the turnover rate and the catalytic efficiency, what we noticed is that SARS-CoV-2 enzyme was twice as fast as SARS-CoV-1. So it's really quite interesting. And this has nothing to do with glycobiology, but I'm just popping this in here. Okay, so moving on to inhibitors. And one of the reasons I bring this up is this is a drug we worked on at the beginning of the pandemic, and it's related to the feline infectious peritonitis. And this was an inhibitor to treat cats who had coronavirus infection. And so in the overall paper, we supplied them with this GC376. So this is an inhibitor that many people use to demonstrate inhibition in cells, and it's an extremely potent inhibitor. And so some derivatives of this we've worked on, but we're not focusing on this as much anymore. But we did do IC50 studies and crystallography studies, and we showed that the potency, we were in the nanomolar potency, so 190 nanomolar. But the crystal structure showed good hydrogen bonding. So this is a cysteine protease, and we see a nice covalent reaction with this, and that glutamine in the P1 position is hydrogen bonded nicely with residues in the active site pocket. But the P3 position was not really well oriented. So then we moved forward and, skipping over a few years of work, we developed different inhibitors with different warheads. So this is an AMK, an acyloxymethyl ketone, or a nitrile warhead, very similar to that found in nirmatrelvir and Paxlovid. And when we look at this with the indole group in the P3 position, we can start to see more hydrogen bonding, and it kind of explains why we start to see, increased potency here. So our potency is now in 19 nanomolar, very similar to nirmatrelvir found in Paxlovid. So our drugs were getting more potent, sorry, I'm gonna go back to here. This is also just a really interesting chemistry notion from our derivatives, that when we have a 5-member gamma lactam ring versus a 6-member ring, what we see is 40 to 13 nanomolar. So just adding an extra carbon in this bulky ring can just change the potency of this inhibitor so much. So the chemistry is really cool with all of these studies. So this is where we ended up. We ended up with some inhibitors that were really quite potent, but then we had some issues with ADME and pharmacokinetics. So we wanted to, before we moved on to animal models, we wanted to improve that. And that's essentially what we did. So we have now made a new derivative, which I'm not going to go into details about, but when we compare it to nirmatrelvir, here we're looking at the half-life of the inhibitors in the blood. And we can see that our new derivatives are starting to have a longer half-life. So we go from 57 minutes to 83 minutes. So this is really ensuring that the drugs that we develop that are intended to be oral will be potent. Okay, but to get to, with that background in mind, to get to the heart of the story, so over time, we've noticed that we've had some changes in SARS-CoV-2 evolution, and these really actually have different arms. And one of the reasons I have this phylogenetic tree here is just to show how distinct they are. But my graduate student, Amelia, who's the first author in this paper we just published this year, she looked through the GISAID website, and this is a website that collects clinical samples from people who have had COVID-19, and she looked through all of the samples at different time points. So she looked at alpha, beta, gamma, delta, and Omicron collections that were sampled, and we looked for changes that occurred in the protease. So as I mentioned, you've heard lots of changes about the spike protein over time, but the protease also changed over time. And so we wanted to examine whether or not this has an effect on the inhibitors. And so what you can notice just from this graph with the frequency of occurrence in darker purple, we had a lot of mutations occurring in the alpha phase as well as the gamma phase, but less in the Omicron phase. So that's kind of where we are now, a little extended from here. And this proline to 132 histidine is really quite prevalent. This mutation also occurred like in beta to some degree. So it's really quite interesting. So, but overall, there are 31 mutations that Amelia characterized. So I'm going to talk about that study today. And so one of the first things we did is we looked at this according to what region in the protein they were located. So they were either found in the active site region. So there's 4 listed here. This is behind the active site. So the active site is here with the cysteine. So this is behind, which would influence substrate recognition. And then this protein forms a dimer, and there's a motif in the middle here that helps solidify that dimer. And there were mutations found here. So we thought these were ones that might influence activity, and we decided to look at this further. So Amelia did assays to measure catalytic efficiency. So using a fret-based substrate, she measured activity and compared it to wild type. And so going from N to C terminus, and including a double mutant here, which is the K90R and the proline 132 his, what we can see just from looking at this graph is that there was changes over time. So as we have different mutations, and for some we see a decrease in catalytic efficiency of the protease, and for some we see an increase in catalytic efficiency. I will point out for Omicron, we actually see a decrease in the protease efficiency. So what this means for infection, it's really quite hard to say. So one of the things we could say is, if you've had COVID more than once, maybe it was really bad the first time. And maybe it was because it was an alpha strain and, the activity of the protease was here and more virus particles were being made. One of the things we haven't followed through with is kind of examining this. But after looking at catalytic efficiency, so I will point out too, there is also one mutation here you might notice it has almost no activity. So this is probably an isolate collected from a person who had multiple virus particles in their system, which does occur as well. We also looked at substrate specificity, and we see that this is using a proteomic analysis, and we see that there was some changes, even though leucine is preferred in the P2 position. We started seeing some other residues starting to pop up, more hydrophobic ones, which was interesting. Okay, so then we looked at the crystal structures to examine how some of these changes could occur. In particular, for this glutamic acid to switch from an asparagine to a lysine, like what were the changes? And so if we compare this Lu50 to Phi change, what we see is the active site, the subsite 2, S2 pocket, starts to open up with this mutation. So it gives a little bit more space and allows the substrate to bind more readily. So we have enhanced activity. And then we have a decreased activity with this one because there's a new hydrogen bond being established and is blocking off this pocket. But in contrast, for this lysine, it actually opens up the pocket to some degree. So the crystallography can really inform us on this. Okay, but getting to the glycans. So now that we have identified that there's changes in activity of the protease as the virus has evolved over the past few years, we really wanted to know whether or not this would have an effect on the host recognition and the host defense system. So I introduced this slide already, and we have, just to remind you, we have the 2 carbohydrate recognition domains in galectin-8 that is supposed to be responsible for sequestering endosomal and lysosomal vesicles for degradation. And so what Amelia did is we purified galectin-8, and here you have the enzyme alone, and she incubated galectin-8 with all of the different variants of concern main protease. And you can see this pattern of cleavage that occurred. And using the lower bands, so with the white arrow, she integrated the densitometry of these. And so what you can see compared to wild type, for some of these mutations, we actually see more galectin-8 being cleaved. For some of them, we see less galectin-8 being cleaved. So what does this mean? So if more galectin-8 is being cleaved, that means it could be potentially dampening the host response to the virus particle. In this scenario where we have less cleaved, we could envision that your galectin-8 is going to be intact. You're going to have a robust response to this infection. Now, Amelia then went further to, instead of this, so this assay is just a single time point with a single concentration. So our lab is an enzymology lab. We wanted to look at rate of cleavage because that's more precise. And so Amelia did this time study where she takes different galectins with or without SARS-CoV-2. And we have a control here of a non-catalytic enzyme mixed with galectin 8 to show that all the cleavage we're observing is indeed from the protease. A 2-hour time point was selected and the reaction was stopped with this feline drug that I introduced earlier. And then she was able to measure the activity over time. And with this, she could calculate the rate, the initial velocity. And indeed, what we do see is some of these mutations cause galectin to be cleaved with a faster rate, and some have a lower rate of cleavage. So the rate is important because it's not just a single time point. Now, and so this is the band that was integrated for densitometry. Now, the next thing we wanted to look at is when we looked at some of these gels, we know from the overall paper, they predicted that galectin was just cut in the middle, like there's just a single site. However, we noticed there's a whole bunch of bands here. So we thought, well, let's examine this further. And so we incubated galectin again with a molar ratio of 5:1, and then we stopped with the inhibitor, and then we did mass spectrometry with the Julian lab here at the University of Alberta. And we were able to show there's actually 3 different cleavage sites with galectin. So one at the linker in between the 2 carbohydrate recognition domains. And at both of the termini, there's some small changes. Now, if you recall when I had the slide up about what galectins do and how they act, we know that the carbohydrate recognition domains are really important for recognizing these glycans, and the dimeric nature is important. And so there's a couple that have this dynamic nature. Actually, before I talk about that, I'm going to actually show a comparison here. So here we're comparing the galectin cleavage by SARS-CoV-2 compared to SARS-CoV-1. And what you'll notice is the cleavage pattern is relatively the same. So this shows that galectins likely are the target of some of these viral proteases, especially the ones that have severe effects, reminding you that SARS stands for severe acute respiratory syndrome. Now, I talked about other galectins that have tandem repeats. So another one of this family is galectin 9. So galectin 9 has 2 carbohydrate recognition domains joined by a flexible linker. And so when Amelia did a similar study with purified galectin 9, we importantly did not see cleavage of this. So this shows that there is some specificity that is happening, and galectin 8 just happens to be recognized by the viral protease. Next, we wanted to see whether or not the cleavage of galectin has an effect on cytokines. So this is in collaboration with Dr. Elahi's lab here at the University of Alberta. And we looked using ELISA whether or not there was secretion of TNF-alpha or IL-6, interleukin 6. And using ELISA assay, we compared no galectin, the full-length galectin, which of course showed stimulation of IL-6 and TNF-alpha, but the truncated versions did not. And this is with PBMCs. So this shows that the cleavage is really having an effect on the cytokine release in cells. Flow cytometry was then used to identify what type of cells was used, were secreting the cytokines. And so this analysis clearly showed that CD14, which is a T cell type, was responsible for this galectin-induced cytokine secretion. And then also B cells were responsible. So CD19 B cells were also secreting TNF-alpha and IL-6. So this shows that there's some physiological response to cleavage of galectin 8s in our cell with virus infection. And so, like a disclaimer here, I'm not a glycoscientist. Like, I like the keychains and I'm really learning quite a bit. But you know, this is a really fun aspect of our lab and where it's kind of growing, and we've really been enjoying working on this. To kind of fill in a loop, a circle of what we've done, one of the big questions that we asked is, since the protease changed over time, we see some changes with cleavage of substrates, whether or not the inhibitors are still potent. And just to end the talk on a good note, a positive note, some of the protease inhibitors are still potent, which is really great. So we compare one of our inhibitors along with nirmatrelvir found in Paxlovid, and we still see on the micromolar ranges we still see low nanomolar potency for all of the, compared to the wild type, all of the variants of concern. So I think this is really positive that the protease is still an important inhibitor. And importantly, we know that these protease inhibitors also inhibit galectin cleavage as well. So when they inhibit the main protease. So where are we going with this now? So we know here I have this phylogenetic tree of different coronaviruses, and they fall into 4 different genera. We have the alpha, the beta family, the gamma, and the delta. And so in red are the common cold, in red here are the common cold coronavirus. And here in the beta subclass beta is the SARS subgroup. And so one of the questions we have is whether or not some of our inhibitors inhibit the main protease found in the related coronavirus families. So it'd be nice to have an inhibitor that works against the common cold. And so we're examining some of the different common cold coronavirus like 229E and L63. And what we know is the sequence similarity is it's very similar, in particular with the active site region. So there's a high degree of similarity, especially with the beta coronaviruses with beta and alpha with the alpha. So you see the alpha are kind of very related to the alpha. So we're really quite optimistic about this. But we also plan to look at galectins. So what is the effect of the common cold and some of the other coronavirus main protease on galectins? So my student Amelia will give a talk tomorrow about this. It's really exciting. She's got some really cool preliminary data, so I'm excited to hear that. So currently, right now, some of our new protease inhibitors, we're assessing the efficacy and potency in mice with the Terrell lab, and our lab is we actually worked on galectin before SARS-CoV-2 with Dr. Elahi, and so we've kind of continued this collaboration, and our lab has already synthesized and overexpressed and purified galectin-8, galectin-9, and galectin-3. So we're starting to dig deeper into the galectins and their effect in virus and host response. And we want to look at the physiological response of this more in cells, like what is happening in the cell and how this can affect infection. So with that, I'd like to thank the members of my lab who worked on this, especially Amelia, who completed this big paper in her 2nd year of grad school. She had support with Jimmy doing crystallography and Elena doing lots of the inhibition assays as well. And we had an undergrad on the project too who looked at off-target effects. It was pretty cool. We collaborated with the Young Lab for crystallography efforts, mass spec. A lot of the chemistry support is just superb. Our proteomics for specificity was done with Marcin Drag's group in Poland. And then we have another project with ECM-WINA that I'm not going to show here, and our synchrotron sources. And I just want to talk about all the funding agencies, just been phenomenal. And more recently, we've been funded by SPARC here, which is funded here at the University of Alberta to work on the common cold work. And with that, I'll thank you for your attention. I'll be happy to take any questions.
Questions? In the back.
Thank you, Joanne. Very interesting talk. So have you looked at the specificity of the proteases on other viruses, or is it just a SARS-CoV-2 specific for example, for GAL8?
Mm-hmm. No, that's a great question. So there's this class of protease, which is a chymotrypsin-like, is also found in, besides coronavirus, they're also found in rhinovirus, picornavirus. So there's a lot of related ones. And so we're just starting to work on some of these. So hand, foot, and mouth, like Coxsackie enterovirus, norovirus, like gastro kind of flu. If you have kids, you kind of know that a lot. But they all have amazingly such a conserved active site. So I wouldn't be surprised if they also affected, like with glutamine in the P1. And so there is a glutamine. There's actually 2 glutamines found in the linker in galectin-8. So it's going to be susceptible to all of these proteases as a recognized glutamine. We all feel so bad when you get a cold. It's not just, and you feel achy through your whole body. And so likely this is an effect of that. So the potency or how strong it cleaves is important. So we're starting to collect more proteases in our lab because the pandemic is sort of over. And, it's been exciting to look at the different classes and their different strength of cleavage. And this is something Amelia will be working on. Yeah.
Thanks, Joanne. Not a glycobiology question, so you can relax.
Good.
Recently there was that Nature paper that said Paxlovid is increasing the evolution of the virus. So I don't know anything about this. Can you tell us, was this expected all along? Is this a big concern?
Yeah,
Can you
so
speak to that?
we saw that paper. So there's a lot of discussion about fitness and how resistance can occur. And actually, in one of the papers, they actually say there is resistance to Paxlovid. But what they notice is that there's just a slight decrease in potency. But I would say that's very different than resistance, where resistance would be it's not working at all. So there are some changes that could occur. So one way to potentially combat this would be include remdesivir and the protease inhibitor together in some combination therapy. And so I'm kind of surprised that hasn't been done yet, but then Gilead and Pfizer would have to play nice together and kind of work on that. Yeah.
Yeah, hi. I can't tell how much I appreciate the painstaking nature of your work, and we need people like you to actually develop these new inhibitors, and it's just astounding. What I'm really excited about is the physiological response that was reported. Do you know, is there a good model, a mouse model for SARS-CoV-2 that you could actually test this in?
Yeah, so there's a couple of models. So people use mouse models. There's some virus it's a mouse adapted coronavirus. So it binds more tightly to the mouse receptor. So that's one mechanism. So in the MAB system we have here at the University of Alberta, Ryan Noyce is working with that. So I haven't talked to Ryan yet, but also the Terrell lab is working with like standard K18 mice. And then there's also hamster model and ferret model. And so there's a whole bunch of different models. As you know, mouse models are quite expensive to also work with. And, the funding for pandemic is kind of going this way, which is fine. But I think that would be a great way to really look at the host response and galectins to kind of move after we do some models in cells, which we can do just in our lab, because we're not set up to work with mice, but there's lots of people here at U of A that are. Yeah.
There's a GAL8 knockout mouse, isn't there?
I think so. Yeah.
We actually have a question online, so I'm going to come
great.
back and read it. So does the inhibitions, this is from Rajesh Kumar Gupta, does the inhibition of galectin-8 cleavage by proteases improve the direct killing of SARS-CoV-2 by galectin-8?
Yeah, so I would imagine, inhibition of cleavage, you would get of the protease. So when the protease is less active with the inhibitor, you are going to see less virus particles being made and more intact galectin that will be present. And so that means your body will be able to respond faster to the infection, and you will be making less virus. So all of us have had infections where you just either get super sick, or maybe you have multiple people in your household who are all sick at the same time. And you're looking at your partner saying, why are you not sick? And I'm so sick. So everyone has this different response. So I think the inhibitors will really help lower that threshold and make you less sick.
Other questions from the audience? Aha.
Thank you, Joanne. Question about your E47K, if I remember that mutant correctly. You introduced it and you showed quite high activity to begin with, and then you showed a corresponding increased cleavage, and you made some innuendo at the very beginning, and I'm wondering if you can just expand on that for us. I was trying to follow the story, and I don't think I quite got that.
Yeah, okay. So one was found with the alpha and one was found with the beta, and one had higher cleavage and the other had less cleavage. So I guess the correlation there is with different waves, there were different levels of symptoms that came with that. And so this is, of course, just a speculative comment, and that's why I kind of don't delve too deep into it. But you can imagine when you're starting to see more cleavage, like with that alpha, you're seeing more virus particles being made. But when it switched to the asparagine and there was less cleavage, you're going to have less virus particles being made, potentially less galectin being cleaved. And so the correlation there is like with different waves, we saw different manifestations of the disease physiologically. And the correlation was that we're starting to tease that apart to some degree. It's not perfect. And of course, because we didn't do the animal studies to really say, oh yeah, well, that's resulting in like a severe cytokine response in a mouse. But we can surmise just from like looking at the data. Yeah. Thanks.
Okay. Other questions? I have one. So I was just curious, so thinking about the galectins as they induce this response, beyond galectin-8, do other galectins also induce this response? And how specific is it, in terms of the cleavage for galectin-8 versus other linked lectins where this dimeric nature might be important?
So galectin-8 is definitely upregulated in this response, but we were talking to our group at lab meeting this week because sometimes when you join a field, you have this like boxes or files and files of paper to read, and there's hardly any papers on this. So we're just like, especially related to infection and immunity, and so the virus response. So in cancer, we see it. So it was on your slides earlier today, like the first speaker, galectins are upregulated in cancer, and we know that. So
yes,
there's not a lot known.
Yeah.
And so it's exciting to work in a field where like everything is kind of new.
Yeah.
Yeah.
I mean, I think there's some that's known upregulation of, I think, galectin-1 and 3 in HIV. So there are some like associated host response with virus, but not, obviously not with necessarily coronaviruses, but there's a little bit known.
Yeah, I definitely have to dig deeper into that.
Questions we got time. So
I don't know if I understand the whole system yet, but is the viral kind of protease, I guess, working inside the cytoplasm of infected cell actually once it's made released infect another cell? That part I don't quite understand.
So it does. It's the protease is translated in the long polyprotein and it's in the cytoplasm. It's not secreted. So it's going to affect lots of host proteins. So other groups have shown this. There's been lots of proteomic studies to show.
Yeah, there's going to be another naive question. Then does that mean the substrate actually are like with galectin-8? Would it be inside the
correct
cells?
Yeah.
Okay.
So this is inside the cell, and it will take the endosomal particle and direct it for autophagy, for degradation. And so it really helps you clear that virus particle before it can start to replicate and make a whole bunch of new virus particles. So it's one of the first lines of defense.
yes, that's so if just to clarify, so this is as your like the virus particle itself, which is glycosylated, is exiting the endosome and trying to get out into the hole.
As it's trying to get out, but as it gets in, so there's lysosomal damage. And I think I've read a paper that said galectin-8 is the danger receptor. Like something's in your cell that doesn't belong here, and there's a bit of inversion and disruption of the lysosome. The glycans the external glycans are exposed in the cytoplasm where they're not supposed to be. And so then the galectin binds to them, sequesters it, and pulls it to the autophagosome. So
that's really cool. Other questions? Well, I think that this is a really great example, actually, of why we need GIA, honestly, because it's a talk in which it talks about, work that you wouldn't think is glycosylation related. And yet glycobiology is playing an important role in thinking about the future of where this is going and host response. And so thank you very much, Joanne, for such a great story.